Hello, everyone. Welcome to today's podcast of the Education and Training Committee of the Metabolomics Society. I'm really happy that we have three renowned experts today with us to discuss the matter of metabolite identification. And it's great that we have some people online. So if you have questions for our panelists, please drop them via the Q&A at the bottom of your Zoom window, and we're going to incorporate them wherever possible. I'm very happy that I have my co-host Fidel with me today. So we both will moderate this. My name is Michael. I'm one of the directors currently of the Metabolomics Society, and I'm heading the Metabolomics and Proteomics core at Helmholtz Munich, especially the Metabolomics team. And with that, I would like to ask our panelists to very briefly introduce yourself. So I think most of people know you, but there might be one or two newbies watching this afterwards as well. So please give us your name and where you're from and what your research interest is. And we're going to start with Emma.
Thanks, Michael, and thanks for the opportunity to be here. I'm Professor Emma Schymanski. I head up the Environmental Cheminformatics Group, and I'm also currently Deputy Director of the Luxembourg Center for Systems Biomedicine at the University of Luxembourg. Also advising the Rectorate on open science and research data management. When I'm not doing all of that, I do like to work on the identification of unknown chemicals, especially in the context of environment and exposomics.
Okay, thanks. We go in the way the windows are oriented for myself. So Pieter, you would be next.
Yeah, thank you. And thank you for organizing this podcast. I'm Professor Dorrestein. I'm at the University of California in San Diego. Just like the rest of the panel members, I really enjoy looking at mass spectral data, but particularly in the context of annotations. What my research is particularly interested in is trying to understand how the chemistry associated with microbes in particular. And we manage a resource called GMPS that the community uses to capture the knowledge associated with metabolomics data.
Great. And Rick, please, up to you.
Brilliant. Thanks, Michael and Fidel, for organising this. Really looking forward to it. I'm Rick Dunn. I'm Professor of Analytical and Clinical Metabolomics at the University of Liverpool in the UK. I'm also President of the Metabolomics Society, so I better not do anything wrong during the podcast. And my research really is focused around two areas. One is metabolite annotation identification and improving our knowledge within those aspects, but also tools within there. And then the other area is really focused around personalised medicine, stratified medicine in humans. I apply only LCMS and GCMS. I don't apply any NMR spectroscopy.
Okay, thanks. And with that, let's directly jump into the topic just to get a bit familiarized with it. What do you think? What are the most commonly applied analytical technologies in untargeted metabolomics? And Emma, if I'm saying metabolomics, I'm including all the small molecules, so even environmental exposure and everything else. And why don't we start with you again?
Yeah, I was going to say Rick almost answered it in his introduction. So I guess currently the most commonly applied ones, also for the exposomics, is LC, so the liquid chromatography, mass spectrometry. We do see both reverse phase and helix becoming more common. In the environmental world, the helix is still catching up a little. So this I see definitely more common in metabolomics. We are seeing a lot more GC. Obviously, in metabolomics, this has a longer history. For us, when we've done water-based contaminants, GC has been less useful. But now with the higher resolution GCs coming up, we're seeing this as well. We're seeing some ion chromatography mixed in. And like Rick said, all of us on the panel today don't necessarily do terribly much NMR, but we do see a lot of NMR happening as well. And certainly from the environmental point of view, this has mainly been a sensitivity issue. We get a lot better sensitivity with the mass spectrometry. We are seeing increased ion mobility coming too, which is very nice.
Cool, great. Pieter, your take. Anything to add?
For the analytical technologies, no, it's primarily LCMS, GCMS, and then NMR. Both largely 2D NMR is becoming really popular at the moment, and I agree. Ion mobility is one of the key additions that will complement a lot of the other LCMS methods.
Yeah. So Rick, you already mentioned in your introduction many techniques. Anything to add on your introductory sentence?
Yeah, so GCGCMS, so comprehensive GCGCMS has been popular for a while within metabolomics. It's sort of, you weren't seeing it a lot and it's coming back again. So the extra chromatographic resolution, and we're seeing more papers coming out with two-dimensional liquid chromatography in mass spectrometry as well. Not routinely applied, but are being applied for specific applications.
Yeah. I think this resonates with all of us that we see we need more resolution in every direction, right? So
I think the one thing we didn't mention so far that certainly now that we're moving into exposomics, there's some metals that are implicated in a lot of diseases. So people are interested in the ICP coupling to the LC so that we can actually get this. This seems to be still quite tricky. A lot of people would like it. The practical reality is not there yet, but if people are interested in the metals, that should also not be neglected.
that's true. That's true. Yeah, right. So what do you believe are the current hurdles holding us back really identifying metabolites, and let's say on a broad scale? And maybe we start with Pieter there, since I mentioned the broad scale already, and know that GNPS goes very broad in application.
Yeah. So for me, I think there's two primary things that are holding back identifying metabolites. I'm sure that can be expanded upon, but one of those is actually the availability of standards. So there's a lot of molecules that people have identified, but the standards aren't even available. So that makes it really difficult to confidently identify metabolites. The second thing is actually making annotations that have been made in the community the underlying data readily accessible for reuse. So if a metabolite is identified in one lab, if you don't have the raw data that's accessible to another lab, you cannot reuse that information. And so this is really holding back identification of metabolites, particularly when you start to go from one lab to another lab to another lab, and really at the repository scale sort of level.
And we're going to have repository scale later also as question, because I think this is a hot topic at the moment. Yep. But Rick, what are your thoughts on this? What is holding us back?
I agree. The standards, if you can't buy the standard or synthesize the standard, it can't go into a retention time library or into an MSMS library. I think that the fact that we can't sequence the metabolome as you can sequence the genome is a big hurdle. We don't know what to expect. And I think maybe later in the podcast, Pieter might discuss, some of his discovery of new metabolites or new classes of metabolites within there. If we knew what we were detecting or what we were routinely detecting, then it would make the job easier, within there. So I think that's one of the hurdles. The other thing is we don't know what the metabolome is. We don't know how small or how large it is, etc. within there. And so there will always be hurdles associated with how accurate or how specific are we in those annotations. And being able to report that confidence to reporting standards, for example, I think is very important as well.
Mm-hmm.
And I think we are hopefully on the larger side, not of small metabolomes, but I believe the metabolome is rather large. And one thing that is neglected quite often is what Emma is working on, the exposome and all the environmental things. So how are things in your field? I guess you also lack a lot of standards, right?
Well, yes, there's a lack of standards, but I think I'll actually agree with Pieter. We actually have a lot of data that's already been recorded that's incredibly difficult to share. And I think one of the things that's holding back is the different, like not separating out the different takes we want to take on the data. If you just want a quick annotation of what's actually already been known, we could probably actually do that quite quickly and you get some reasonable confidence, but then people always want to dig into the unknowns. And at some point we have to let the unknown go, annotate the known and move on with it, move on with what we can conclude. And then, yes, we still need to do the fundamental research on the unknowns, but this is a completely different question. And then here we're really getting to the data availability. What are the ones that are out there? And I think for the exposome, we have the same problem as a metabolome. We know the database is incomplete, and at the same time, it's too much data. But if people would be able to share those annotations, and we also know there's an awful lot of stuff that's recorded where the raw data, like Pieter said, is just not available to us. So people claim that we have the data, but we don't actually have access to it. And this is where things like proteomics and genomics with the forced data sharing made a lot of progress.
Yeah.
Here we've got a much bigger space, much bigger complexity and much less data upon which to make all of the machine learning the thoughts, because we have 119 million chemicals in PubChem. We've got rich data for a couple of thousand if we're lucky.
Yeah. I think my answer was a little bit even more nuanced than that. It's not so even when data is available, and it's the raw data to the experiment is available, and then you have a list of annotations. But there's a couple of things that are often missing. One is the link of the annotation to feature within the data set, because once you make that link, you can reuse it. But also the provenance of how that annotation was attained is completely missing. There's no resource that really captures that. And because that will allow you to double check, do you believe that annotation? And then the other one, it allows you to reuse it so that anybody can take that knowledge that you've created and enable reuse. And that should be fairly efficient once the infrastructure is in place to be able to do that.
Thank you. Speaking from the data, as we're talking about the data, I will ask you, Pieter, what data can be collected or should be collected to assist in annotation or to identify, let's say, using LCMS, maybe start from there?
Yep, no, I really like that question. And this is where I feel like the community can do much better, and it follows up what I just discussed. So let's say you write a paper, you have your annotations, okay, you go to publish. It becomes either a table and a supporting information, okay, here we have a smiles, here we have the name, and here is maybe a relative peak area. And then it ends. If you're really lucky, and this is about 1% of the metabolomics data that gets collected, they actually deposit the raw data in the public domain. So that's a major advance if somebody already does that, so that you can actually go back and look at that raw data. But the dream, and this is a long-term vision that I think the field will need to migrate to, is to make sure that this annotation is linked to the provenance of how that annotation was derived. So if you have an MSMS that is annotated, you say, let's say it's cholic acid. Well, how did you get that annotation? Did you run it with a standard? Okay, then the provenance of the standard, the LCMS of the standard should also be there. And those two should be linked so that you can double check that. If you annotate it based on a reference library match, well, then it should be linking to that reference library spectrum with an identifier so that somebody can double check, okay, that's where it came from. But then that, if you have an annotation, that should also go back to your original LCMS data and say, okay, this is the retention time, this is the exact peak that is there, and this is the MSMS that belongs to that annotation that I've identified. And the reason why this is so important is because now you have all the evidence to say, okay, did I believe that annotation based on the information that I've provided? And then you are able to reuse it. And so if everybody ends up, if we build the infrastructure that way, if we build new MC-Mine tools, if we build MS-Dial modules, if we Metabolite, GMPS, and other resources, if they start to work together to capture that information, it will really significantly enhance what we can do within the context of the community.
Thank you, Pieter. So if I understand, you are saying the need to collect, to link the raw data, the standards to the annotation that is reported. If I go to Rick, in terms of what kind of data spectra elements that you can advise to say for annotation, to start for annotation, these are minimum requirement type of data that you should collect from your LCMS analysis. What would you advise?
LCMS typically can collect at least 3 types of data. One is we always collect the mass of the metabolite feature, but remember that one metabolite can be detected as multiple different features, different adducts, etc. We always collect the retention time, the chromatographic retention time as well. And then depending on how you're collecting the data, you can collect MSMS data as well. And there are different ways to collect the MSMS data. You can either collect it using data-dependent analysis, where you get a relatively pure, specific MSMS mass spectrum, or you can use DIA, data-independent analysis, or SWOT analysis, where the mass spectrum can be a bit more cloudy, a bit more noisy within there as well. So we always collect MS1 data. We always collect retention time data. Sometimes we do, sometimes we don't collect MSMS data, depending on a number of factors, such as if you've got a low intensity peak in your MS1, you would normally set your instrument not to collect MSMS data on that, because you would never collect anything because of signal to noise on there. And then there are other things such as ion mobility. You can think about 2DLC, having two chromatographic retention times, etc. as well. I would say the more information you have on all of those, the more confident you are. Just using MS1 will get you so far. It will reduce your search space down from a very large search space down to a smaller number, but it won't get you to a high level of confidence. So using retention time and MSMS, I think, is really important as well.
Thank you. Ima, you work in environmental matrices. I'm sure there's quite a lot complexities in the samples that you collect.
But the data is the same. I think Rick covered almost everything there.
Okay. So I want to jump to construction of the libraries. How do we use RT, retention time, MSMS, to build the libraries that can help? Is it limited by the availability of the chemical standards? Some, for instance, those who work in natural products, sometimes there is really no chemical standards that one can use. How do we tackle such challenges?
You want me to answer that one?
Yes,
I was going to say, I mean, it's difficult to construct a measured MSMS or retention time library without the standard. So obviously then we are limited by what can be purchased. Although we do know that there are a lot of people producing libraries with rather large numbers of chemicals that we don't have in the public space yet. So obviously these existing standards, it would be useful to have that as open data so that everyone can profit from it. What we do see is a lot of people in natural products, but also the identifications in the environmental, the transformation products where we don't have the standards. You can do actually a very detailed identification, for instance, supporting with NMR, so that the identification is clear, even if the authentic chemical standard isn't available from an independent vendor. And then once this identification is confident, once you have the NMR that actually confirms the structure, then we can also make a highly confident reference library entry, even without there being a purchasable standard. And this is where Pieter said it's so important to get these entries tagged in the mass spectral libraries, because these are the really valuable data. We've actually helped get some new PFAS transformation products into PubChem, into mass banks, so that people can see the mass spectrum, the NMR spectra of some of these singly characterized compounds, so they can be found around the world. And this is where data sharing is extremely important, and also working with the reputable groups to help them get their data out there, because this is where Pieter and I have been working for many years to share open data, and we're always willing to help people add new data to the libraries. So.
We have a perfect question on this already from the Q&A, and here is what's written is, I've noticed that some of the spectral data made available in spectral libraries is poor quality, which affects the confidence of the annotations made. Are there any guidelines that are considered when individuals upload raw spectral data into this library, or is anything accepted? So what's your take on this? Rick, you're already waving your hand.
Having just, with my wife in the lab, collecting data for standards for 6 months across a whole range of kits, I think it's very important to manually go and assess your data. So you can use software to look at the data. It will find a retention time, it will find an MSMS mass spectrum, but not necessarily with high confidence. In some cases, if you've got really high concentrations of your standard, you might see a double peak, for example, and it will report both of those. Well, only one of them is actually correct, for example. If you've got a low intensity MS1 peak, then really I wouldn't be pushing that into a library, the MSMS into a library. So the manual assessment of the data, I think, is really important within there.
Thank you, Rick.
Emma raised her hand.
Yeah. I'm sorry, Peter will also have a good answer to this one as well. I know from the mass bank point of view, we accept several collections into mass bank and we have validation. We can also provide contributors with recommendations. So we've actually had a submission quite recently where we noticed there were thousands and thousands of peaks in some spectra. We flagged it with them and they say they want to keep this in because this is what the data really looks like. So if you're looking to match genuine instrument to instrument, that's what you'll be matching. What we have enabled, this gets back to Peter's point on the provenance and, you know, all the details that you provide with the mass spectra. In mass bank, we have a lot of different tags. So the people who want to use this data can always include or exclude different selections. So like we know we have a lot of really old GC data. It's extremely valuable. It's the only data that's in the open space that we know of. But if I'm trying to create an LC-MS-MS library for identification exposomics, I don't put the GC data in there. So we will basically integrate a library that contains the data that's most relevant for us. If we have an Orbitrap, we will preferentially take the Orbitrap spectra, not necessarily match to QTOF spectra. If you want a broader range, then maybe you have all the QTOF spectra mixed with the Orbitrap ones as well. So a lot of it is sharing the data is really important, and helping people overcome this barrier is for us really important. But also enabling people to choose if they just want the high quality data, if they want all the data, is also very important, so they can answer the questions according to their, according to what they need, basically.
Yeah, I'd love to follow up on this as well. To me, this is a really interesting philosophical question. One, there's no standard for reference spectra deposition, so that's part of that question. So it doesn't exist yet. Not sure if we even should have one. Instead, I think we should have some sort of quality metrics, like MONA has a quality metrics of the information associated with the spectrum, which I think could be used, for example. For me, any reference spectra that has an annotation is better than not having the spectrum in the public domain. What will happen if it's a poor quality spectrum and you then try to match it against data that you're working on, you will not find a match. So you're only going to lose it. You're not necessarily going to gain an annotation because it's a poor quality spectrum. There's also really good filtering metrics. So one of the common metrics of filtering or ways to filter spectra is just like in every 50 Dalton window, you pick the top, let's say 5 ions that are present, and then you eliminate a lot of noise that exists within that. And then if you try to match it against your real data that is also filtered in that same way, you're not going to get too many mistakes. But so, and the reason why I say this partly is because sometimes reference standards are created with materials that are just not available. They needed to run an LC-MS on an extract because the compound is not available, and they're the expert in that particular sample, and they know that that molecule exists. Even if it's a low intensity spectrum, I think it's still important to capture that knowledge so it can be reused. So those are some of the challenges when you start to look at reference libraries. But just like what Rick was saying, it's really important to double check manually. I'm a big fan of manual checking. The GMPS dashboard can now retrieve any file from Metabolites, Metabolomic Workbench, if it's deposited there. It's being billed to read Zenodo. And GMPS masses, so that you can just specify the file, the hyperlink to it, and you can look at the data and the MSMS spectra and so on. So there's a lot of nuances that go into these library creations and availability of them.
Thank you, Peter. From this discussion, I think the Rick tasks for revising the confidence level for annotations. Some of these specifics to be included and the guidelines provided. So there is also, in terms of technologies, different analytical aspects that are coming up. We did mention the STAT, the ion mobility. If I go to you, Rick, how could this aspect of ion mobility really help to improve the annotation?
Okay, so I'm not somebody that uses ion mobility in my lab, so I'm just going to talk about it from a general point of view. And really, it's about adding an extra level of complementary resolution or complementary separation. So ion mobility operates in different ways to chromatography, whether it's gas or liquid chromatography, and so will separate on different properties. And so by using that and using whether it's now a mobility time or CCS, etc., it will provide an extra level of confidence basically within there. Again, it comes to be able to run standards, though actually in silico prediction of CCS is becoming much improved and relatively accurate from what I understand and speaking to others as well. So, so unlike retention time where we can't necessarily predict very, very accurately, I think in ion mobility you can. But somebody else tell me if I'm wrong.
I was going to say, if I can add here, I think this is where ion mobility has been extremely valuable for complex matrices, and it's actually in two ways, and it still depends a little bit on how the instruments themselves operate. So different vendors have different stages where the ion mobility is applied. But what we actually found in working with complex environmental samples is the cleanup that the ion mobility provides by separating the fragments out of the complex matrix has actually, in cleaning up the spectra, really helped improve the identification of contaminants in very complex biological matrices, for instance, above and beyond what actually the ion mobility alone provides as the additional dimension. So there was a fantastic study we did in Sweden where he really showed the improvement of the MS-MS match values across the board of the entire identification was really significant. And of course, like Rick said, we also get the extra dimension. So it does correlate with mass. So the resolution of most of the instruments we have is not sufficient to always tell between very, very similar isomers. But because you get the collision cross-section gives you the area of the molecule, you can tell between very long molecules and rather more compact structures. And like Rick said, the predictions are actually getting very good. So we've been working with CCS base. We actually have this calculated for all of PubChem Lite, which is a subset of PubChem. What we do know from the exposomic side is some of the areas for which we have less data are still not as accurate as we'd like. And this is, again, why we're facilitating researchers to get as much data in the open space as possible so that we actually get enough data for the predictions. But it's getting actually very, very nice and very good to use. You can get some very fascinating trend lines according to different groups with the halogens as well. They separate out very nicely from the matrix.
Emma, can I just ask, what about doubly and triply charged species? Do they separate out from singly charged species as well?
And this also depends on the type of the instrument where the separation actually happens. So yes, you can actually see it either on the ion mobility side or you can see it beforehand.
Yeah.
So yeah.
Thank you.
Yeah, we do use ion mobility now in the lab, and it's truly complementing sort of chromatography. Very close structural isomers where you just have a change in, let's say, the stereochemistry of hydroxyl group. If there's more stereocenters in your molecule, you have a chance to potentially separate it. Or leucine versus isoleucine conjugations, for example, you have the chance to separate it. It's the reason why I say it's truly complementary is because some things you can separate with ion mobility, and sometimes you can separate it with chromatography. And I don't always fully understand why which one gets separated by chromatography versus ion mobility. But the complement really adds a fourth dimension to the mass spec to further resolve your annotations.
Great, great. So what I'm hearing right now is that we need more reference standards. So I think one take-home message is how can we get natural product chemists and organic chemists interested in more metabolomics synthesizing stuff for us. But you already mentioned in some of your aspects already some tools like CCS-based for prediction. So I think this is one big elephant in the room. AI, machine learning, right, is everywhere. It as well in metabolomics. So I would be curious on your take. What is your take on the AI machine learning approaches you can utilize to predict or validate metabolite structures? And maybe we start with Emma.
Well, I was going to say it's AI is obviously a new trend, but we would have to say that programs like Sirius and CFMID have been around for, I think, a scary number of years now. I think it's over a decade. And these have been using machine learning right from the beginning of their existence. So it's been around for a while, and we know that Sirius and CFMID could definitely make a distinct improvement on the bond disconnection approaches. I think what we still see and what we still see in our data is actually a mixture of approaches is still what we need. So if you combine the bond disconnection with the machine learning tools, we still get an even better ranking. Get back to my original point, what the problem is, we have a chemical space of over 119 million chemicals, and we have rich data on very few of them. So any prediction is very good within that domain and a little bit outside that domain. And all of the molecules that none of the machine learning has ever seen, this is where it begins to struggle, and you will get answers. But what's often difficult with some of the black boxes is then how good is that answer or not? It will give you what it thinks is a very confident answer, but we might not be so aligned with it. So there's a lot of potential, and it gets back to the more data we have, the better they will get. I think one thing we mentioned, I can't remember how often we mentioned it here, is a lot of it is also about the interpretation. So identification is one, but interpretation within the context is another. And if you would put a ML approach on an entirety of PubChem, you will get all sorts of answers. But if we're actually talking about a metabolome where you've got quite a restricted set of metabolites, then an annotation would likely be higher quality if it's already in the domain knowledge that we know.
Great. Pieter, what's your take?
I think there's one thing that Emma touched up on, which was really accurate, and that is for reference standards that are used for training AI and ML approaches for structure prediction. Those you do reasonably well, particularly if you're okay with just class prediction. So if it's a peptide, or if it's a flavonoid, or if it's a bile acid, for example, it works reasonably well. Are they good enough at this point to truly predict all the nuances of a structure, the regiochemistry of a hydroxyl group, the stereochemistry of a hydroxyl group? No, not yet. What will need to happen before that will take shape is we need more reference data. And just what Emma was saying is there are certain classes of molecules that are completely missing. The other aspect of it is the nuances where you have different adducts and different ion forms that are present in mass spec. So sodiated, protonated, ammoniated, and so on and so on. A lot of these are actually don't have many reference spectra at all in the public domain, and so that also limits the training.
Can I jump into that point, Pieter?
Yeah.
Because I think this is a really important thing. You're faced with the same problem at GMPS. It's easy for us to upload data that's been taken. It's very hard to share data where it's like, we know we didn't get the negative ion, but you can't create a record of something you didn't get. You know, people don't share sodiated spectra because they rarely fragment. Everything is lost on sodium, and it's very rare that you get a fragment. So this is severely underrepresented in mass bank because nobody would take the spectrum of it because there is no data. So this is one resource that is missing. We know implicitly for the Earwald collection in mass bank, we recorded everything in positive and negative. So if there is no negative and there's a positive spectrum, we know that thing actually did not ionize in negative mode. But that's implicit data. There is no record saying this spectrum did not, or this did not ionize in negative mode. How can we actually share that non-data?
Yep. And then, so beyond just for structural annotations, I think I think there are real opportunities, and there's some examples that are existing in the literature where AI and machine learning can help retention time prediction, and particularly retention time order prediction. So there's opportunities there for sure to complement, let's say, AI and mobility, or even CCS values. There's now AI and machine learning approaches to predict some of the CCS values. So there's room for AI and machine learning. There's still a long way to go to make that routinely applicable. One area I feel like it's been useful in our case is actually substructure prediction, so that you create, identify sub-regions of an MSMS vector that may belong to a specific substructure of a molecule when you try to elucidate the structure, and that has been useful.
Great. I don't want to jump over you, Rick, but I think the next question is the perfect one for you on this. But before I'm asking this one, I would like to ask our audience, please, if you have any questions, use the Q&A box. We're going to try to squeeze in your questions wherever they're going to fit. So please, the people are here to answer your questions. If have, please post them in the Q&A. And the one for you, Rick, since I know you are very, very active in the Metabolomic Standard Initiative, is actually how can these in silico predictions, CCS, retention time, structure, whatever, actually fit within the scheme, or do we need to redevelop the things?
There's two aspects to that. There's one, there are reporting standards for metabolite annotation identification that were published in 2007, and I'll be honest, next to nobody is using them to report the confidence of the annotation or the identification. So the annotation is a lower confidence, and identification is a higher confidence. And most, to be honest, of what we're doing, according to those definitions, we are doing annotations in metabolomics for a lot of the data. So we have to always report the confidence on that. Those reported standards report four levels, very basic levels. That's a very good starting point within there. With what, and Emma's been involved in this as well. Emma was working on the paper this morning for this, finally. We have developed another set of reporting standards for this, which is more, there's more resolution in there, and it's based on the confidence of the chemical structure that you're applying. And so a level 1 would be, could only be NMR, for example, if you're doing NMR, 2D NMR on something. And so there will be new reporting standards hopefully available this year that we'll be making publicly available.
Great, thanks. Somebody else maybe has an opinion on this? It was just a perfect question for Rick.
I was going to say, are you after an environmental take on that? Because then I would jump in and say in the environmental world, we've had a 5-level system for a while. So this one we released in 2014, where we've also hooked in the confidence on the structure with the language used to communicate the level. So level 1 is where you actually have the reference standard, so you can confirm it with the measured data. Only one structure, so it's that one structure and you've confirmed it with the standard. Level 2 is the same, you only have one structure, but you've either got there with a spectral library match or a diagnostic fragment, and diagnostic fragment has been misinterpreted a lot, but there we really mean this diagnostic as in you can eliminate all the other structures. And what most annotations are, if there's more than one structure, is effectively a level 3. And this is where it comes to when working with in silico predictions, most of these will end up with a level 3 because you've got some kind of data to support some candidates above another, but it's unusual that you get only one candidate remaining. And then we have level 4 formula and level 5 exact mass. So it's pretty much almost everything is a level 3 unless you've got much higher confident data. If you've got a really good mass spectral match in MONA, GMPS, mass bank, then we get quite a few level 2As. And then once you've got the reference standard in-house, it would then be a level 1. And that would correlate with the identification that Rick was saying. So level 1 identification, the rest are annotations.
Great.
Yeah.
We have a question.
Sorry, we actually have, very exciting. There's actually a group in the US on the environmental side developing an app where people can actually put in their data and hopefully get these levels out and start to see what evidence belongs with the confidence. I think what we've noticed over time, people tend to overestimate their level. So it's like, I'm pretty sure it's this, so I'm going to give it a level 2, but actually it's often a level 3. So
agreed. Also did it from time to time. So I think we're all just humans at the end, and we
Yeah.
like our data and our annotations.
So
But we
we're
have-
curious how yeah,
Yeah.
how we
But
can
we have a
organize this.
perfect question on that, and it is: are we shooting ourselves in the foot? By the way, I really like the wording. Are we shooting ourselves in the foot by developing machine learning and AI predictive tools that rely so much on similarity scores, when it's quite apparent that similarity metrics are subpar at best? So, anybody has an opinion on that one? Up to everyone.
I don't know what is meant by subpar. I would need some clarification. I think similarity metrics, if you use them properly, you can rely quite a bit on their annotations. Now, if you want to be greedy, yeah, you're going to get a lot of errors. If you're going to be more strict, you're going to get less errors. But the AI and machine learning approaches are not dependent on those metrics most of the time, because they are developed independently of any of those metrics. They're developed based on reference data, training data, and then whatever training is done, that's the model that's being built that is then reused. And at that point, there's no more similarity metrics that come into play, or at least not the traditional ones. Now, we do often compare, and when you develop an AI or a machine learning approach to elucidate structures, to what's let's say a modified cosine or a cosine correlation will find, or entropy correlation will find. But the algorithm itself, that the AI or a machine learning algorithm, is independent of that.
Okay, thanks. Fidel, do we have more questions?
No, yes. There is indeed efforts from the discussion. There's efforts in improving the annotation. And I think with the AI, ML-based tools, people already are getting to annotating the spectral data. But one can still ask a question: how large are the metabolomes? It's really complex questions because different matrices, but what can we say about the dark matter, dark metabolome? I will go with Emma first.
I was going to say, I'll expand from the metabolome to the exposome. No, I think, so out of the databases we know, I mean, HMDB is already on the over 200,000 metabolites. So PubChem Lite for exposomics. So this is a collection we try to, of PubChem that we try to collapse down to make annotation and exposomics at least reasonably fast. This is 400,000. We know, of course, these are incomplete. And I think if you're even starting on the lipids, I mean, we know lipid maps is also well over 400,000, and that doesn't even have all of the isomers. So this gets back a little bit to what Pieter was talking about with the class annotation. We have, for many classes, we would probably have one or two of the most common representatives, but not necessarily all of the combinations. And if you start going into those numbers, I think it would be mind-boggling. So all we know is, I know PubChem is 119 million, and I know there's a lot of data that's not in PubChem. And we know CAS is, so out of the exposome, it's 219 million. We also know if we would take biotransformer to transform even, the regular transformations that we see, conjugations and whatever, we can get an expansion very quickly in a few generations to the billions. So the numbers are scary. But this also goes back to the question, we know some of it could be in source fragmentation. We know some of it is likely going to be analytical artifacts that we don't understand yet, but the community understanding is getting there. But we also know that a lot of it is actually data that's not in the libraries yet that would be really nice to share, so we can get the experts to annotate this, share the data, and improve the annotation for others afterwards. So we're seeing many causes for the dark matter.
And Pieter?
Yeah, I really like this question. I think in the end, Emma is right. The world metabolome, so to speak, is going to be in the billions, and so this will, it gives us job security. But let's say we take it one step back and just say, okay, what about what has been detected? I think that is probably just as interesting of a question, and maybe a subset of this even. But currently, if we look at the MSMS spectra across metabolites, metabolomic workbench, and GMPS massive, 2 months ago, we just surpassed 2 billion MSMS spectra that are available in those 3 resources. These are all the public data that has been deposited where people have done LC-MS data. Let's say if one molecule is represented by 20 different ion forms, so in-source fragments, sodiumatics, multimers, and all those kind of things, you still are left with a pretty large number, 200 million. But let me just take the subset of human data in account. My guess, so HMDP, as Emma said, it's 200,000 to 300,000. Most of those are predicted. It's only 30,000 of those that are observed, reported to be observed, and the rest are predicted today in HMDP. It's going to be at least an order, maybe 20-fold increase in number of molecules that have been detected in humans. So we only have a 14% average annotation rate at the moment. So there's a lot of discovery that remains when you look at these kind of numbers. So let's say of detected molecules in humans, it's going to be 3 million to 6 million or so.
Just to give a number to add to mine. So in PubChem at the moment, spectral information for 1.6 million chemicals, of which about 466,000 is NMR and 1.2 million mass spectrometry.
Yep.
And that's, again, only a fraction of what's out there.
Correct.
Can I just add, Peter's came across with a really good point. It's what is potentially there and what is routinely detected are 2 very different things. And the size of what's routinely detected is much smaller, or what is routinely reported in the data repositories, for example, as well. And that is in the number of low thousands that are routinely reported. Watch this space, you'll find out more about that later this year.
I have here actually 2 questions. So the first one is now a bit more provoking, and it says here, fascinated by the annotation levels and specificity. Not to say we shouldn't do it, but the biologists who will be working with the data might have the knowledge of the fine detailed chemical structures. Do we risk creating confusion for those if we get too detailed? Even something as simple as R or S designation will confuse some biologists. So I have to tend this question resonates with me a lot because I'm getting this question on a daily basis, actually. So, and I see Pieter already nodding, so maybe you're the first one to answer this.
Yes, not only that it's confusing for the biologists, it's confusing for everybody. So this is a real struggle. Somehow we need to come up with a way that we're going to create some sort of layered reporting where you have, I mean, even if you get a match to tryptophan, it could be L or D tryptophan. But it could also be the beta version where the amino group is moved over by 1 carbon. This does exist in biology. So even if you get a tryptophan annotation, it could really be 4 structures. And so unless you have additional evidence, whether it's the nitrogen is on the alpha or beta carbon, you in principle should not be able to distinguish those. And then you also have the DRL configuration. And if you don't have that information, you couldn't, in principle, you should not be able to provide that unless you use some other priority. Okay, L form is more common. The L form with the on the nitrogen in alpha position is more common, and therefore we're reporting that. So we need to be able to provide the level of information and the amount of confidence. And this is really where it comes back to provenance of how that annotation was derived, so that somebody who really needs to look and do follow-up experiments where they may spend out another $1 million as a follow-up experiment, they need to decide how much knowledge is known about that particular metabolite before they proceed. Because the experimental design will be different for a biologist if it's 1 molecule versus 4 molecules that they have to test. But if they need to test 4 molecules, we need to convey that information, is my opinion. But it will also lead to a lot of confusion, and there's no easy way around it beyond really clearly reporting what you found.
Great.
Can I
And Rick
add-
raised your hand. Yes.
Yeah, can I add to that? What Peter's saying is, don't just report the top hit from an MSMS mass spectrum, report everything that it may be. And if it's a list of 5 or 10 things, that's absolutely fine. The amount of papers I review, and there's a single match and it's just used MS1 data, and it's like, they've labeled it as citric acid. Well, it could also be isocitric acid, and they've not defined. So just everybody, please, you'll know if it's me reviewing your paper because it'll be the first thing I ask, but please make sure you report everything that it could be. And I go to the level, I don't report the different stereoisomers, etc. I will just say tryptophan or beta tryptophan, etc. To try and ease the understanding as well.
Yeah. Rick, I fully agree with this. One of the things that we probably need is to also push some of the annotation tools to push this more.
Yeah.
And it includes GMPS Massive, it includes XCMS, it includes MZMine, it includes Mona, it includes MS-Dial. We really need to do a better job as a community.
Yeah.
So it's an interesting problem that we've come across going back and forth on some annotation, because mass spec often can't tell the stereochemistry, and for some of the standards we actually get them as racemic mixtures. So you don't get one or the other stereoisomers. So we've recorded them and reported them as what we bought as the standard. And if you want to get even more complicated, we've actually got the salt form, but of course we only record the spectra of the neutral form, not the salt. So provenance is also not always trivial. But then when we've been providing annotation, people have tried to correct our annotation back to the most popular form, but we didn't actually have data on the most popular form. We had data on a mixture. And so then you also get, when people are trying to merge annotations and get everything in one form, we don't always have the data at that level. So this gets back to exactly what Peter was saying. We actually have nuanced levels of what we can provide. And sometimes we don't provide the stereochemistry because we can't determine it. And then people will flag our results and say, but this is wrong, this has stereochemistry. And it's like, it might do, but we don't know it. So we'd rather report it without it, because then we can only report the knowledge that we have, and not the knowledge, guess the knowledge that we don't have, that we have no evidence for. It's not easy, that's for sure.
Great. We have another question here on use of standards and the use of surrogate standards. So somebody would like to hear your opinion on surrogate standards. And the idea is basically one lab identified something in one matrix. We already covered this partially, and to verify in another lab, we're going to send over the matrix just to re-identify a thing. So what is your take on this way of identifying or sharing reference data? Anybody?
My immediate question would be, how are they defining a surrogate standard? Because I think this might be different terminology between metabolomics and my community.
Yeah, so they said it's, for example, plant extract or something. They say, okay, you share an extract. You say, okay, look, I have measured this. I have identified these things. Here's the extract. Go run it on your system.
Or maybe to elaborate, for instance, if you know a specific plant that contains specific compounds that you have once annotated, and you use the extract from that plant that is known to contain that specific compound, and you annotate your unknown different extract. What do you think? It's not a pure standard, it's not that bad, but let's say this plant can produce this specific caffeine compound, and you have a plant extract, and you use those extracts from that plant, from which that specific compound is present. Can that serve as, that's what Understand meant by surrogate? To what extent confidence and
Yeah.
Peter.
the case
I'm happy to
especially
answer that
Okay.
partly. So GMPS in many ways is part of the foundation of building the libraries within the GMPS is that people who are experts in their specific domain, if they are experts in their particular sample, let's say you work with Arabidopsis, or if you work with Bacillus subtilis and you know the key molecules that are really present in those organisms, we wanted to capture those reference spectra because the compounds may not be commercially available. It would take you, a large amount of money, let's say $100,000 to isolate the molecule. That's the sort of scale that we were talking about. And so having somebody provide that annotation would benefit the entire community. And so I feel that sort of surrogate approach is extremely valuable. Another example is let's say you do a drug metabolism study in a cultured some sort of cultured human cell line. You know what drug you added. You see new metabolites such as oxidation coming up. These could be very much used as a surrogate reference because you may not know the full structure, but you can leverage the compound. And I think there's a lot of value in it as long as it's properly used and, again, properly described and documented.
Thank you. So we have a few minutes left. So to really thank you so much for the discussion and the insight provided on metabolite annotation. To end with looking the future outlook, I will give you, each one of you, 5-10 seconds if you can say what do you think the recent development in analytical technologies, software, AI, how this recent development will increase the confidence in the annotation of the metabolites, or what are the future development that you see in those lines that will help in improving the annotation. So I will start with Pieter.
So for me, I'm actually going beyond the improvement of annotation. The usefulness of the annotation is also something that comes into play, and this could also be with unknown molecules, but try to be able to understand, link it to metadata of where these molecules have been found, or what are the sources of these molecules. So to me, that's really valuable. It provides an additional dimension of annotation that's often ignored in metabolomics, so that you can quickly say, that's a drug metabolite, that's an exposure molecule, that's a microbial molecule. That is really one of the areas that I think will be really important over the next decade.
Because I'm going to agree with Pieter on that. They're working on the interpretation, not just the identification, is, I think, really critical. And what we've also been trying to focus on in terms of analytical technologies. I'm excited where CCS and the ion mobility might go, because if SLIM and some of these methods become more routine, they can really provide resolution that might actually be able to separate out some of these cases that we can't yet. And this would be very cool, because this can actually get us then the nuances of the structures that are not yet available to us, but should be soon.
For me, just in 10 seconds, 2 areas. One, the complexity of left spray ionization process and using that for annotation. And then secondly, I know Michael will like this one, is retention time. We don't use retention time enough for a number of reasons, and really we need to be using it more. It's very complementary to the mass spectrometry.
Agreed.
Great. Thank you very much. So we are on the hour exactly. It was a great podcast. We still have a few questions left. So I hope all the panelists agree, for the people that still have open questions, if it's okay to drop a message towards you, email or Blue Sky or whatever, LinkedIn, where you can be found. Please take the chance. Thanks to all the panelists, thanks to all the attendees. So it was very well occupied. Thanks for all the interaction. As most of the people say, when you start a paper on metabolite annotation identification, it's still the bottleneck. And I think it is still the bottleneck, but I think we can widen it up in future a little bit. And thanks for all your efforts on doing this. For all the people attending, thanks for stopping by. Please have a look on all the social media of the Metabolomics Society. There are more podcasts coming up on different topics of the Metabolomics Society or metabolomics in general. These podcasts are available as recording on the MetSoc webpage. So even if you missed a few sentences in the beginning, please, it will be there within a few days. So you can rewatch this very lively and great discussion. And with that, thank you, everybody. Happy that you have been with us, and see you around. And if not online, then please, the registration or the abstract submission will be soon open for Metabolomics Society in Prague in June. I hope that all of you or most of you can attend, and we're going to meet there in person to carry on this great discussion. Thanks, everybody.